So, you know what you're about to get? The poop gang. So get ready. I'm glad you ate, and we don't have to deal with this anymore. But we are going to talk microbiome. And without no further ado, my esteemed colleagues, the Sonnenburg, and their amazing team of Sean Spencer, Amibot, and Sungmin Kim, it's yours.
Okay, it's wonderful to be here. I'm going to lead things off today. I first want to just say thanks to everybody who put this amazing event together. We love doing science with Christopher. We love doing other things with Christopher because wherever he is, there seems to be a lot of really passionate, wonderful people, and today is no different. So it's great to be here with all of you. So this is a great panel of speakers, all interested in the gut microbiome. Of course, we have this complex community of microbes living inside of us that is directly downstream of food and then impacts all sorts of things having to do with our health. There's an incredibly rich community of people at Stanford studying the gut microbiome, but I would say the vast majority of them are not studying food in the gut microbiome. And I'll show you their names in a minute, but for those of you that are just studying food and want more connections to people studying the gut microbiome, come to us, and if we can't help you, we'll connect you with somebody who can help you. Okay, I'm going to let people, so we have to stick to a tight timeline, so we have time for Q&A, and then the open mic, I'm going to let people introduce themselves. We have a great lineup here. These are all the people beyond who's just on stage that are doing microbiome research here. I'm sure I'm leaving people out. If there are people that you know that are studying the gut microbiome and are not on this slide, let me know and I'd love to add them. But we have a growing, wonderful community here. Okay, your gut microbiome. Who knew that poop could be so photogenic? So this is a cross-section of the colon, and in the bottom right, you see all the colorful little jelly beans. Those are all the different bacteria that live in your gut. And then the top left, all the blue area is your your colon cells. And so you can see how close all of these microbial cells are to your human cells, just separated by this thin green mucus layer that we secrete to keep the microbes in the right spot. But this is an incredible interface for exchange of biological information, and it's what most of us think about when we wake up in the middle of the night. Okay, so we've known for a long time gut microbes are important in gut health, they're important in digestion, but they're important in everything. If you screw up your gut microbiome, you're going to impact your immune system, your metabolism, brain gut, we've heard a lot about, neurodegeneration. There's just a ton of things. Basically, if you send ripples through your gut microbiome, that's going to extend through your entire body. And so the question is, can we build a set of tools and knowledge base to impact our gut microbes in a positive way that will then positively impact our health? And it's very clear from all the research that's been done in the field that diet is the way to do this. Like, diet is the way to impact your gut microbes in a positive or negative way if you're not eating right. Another part of our research program studies the gut microbiome across lifestyles, and it's become also very clear to the field that in the industrialized world, we've done a number of things, bad diet, but also antibiotics, sanitation, a lot of things that are very good for things like infectious disease, but very bad for the gut microbiome. So we've messed up our gut microbiome. There's a lot of evidence in the field that this is actually causal for a number of diseases, autoimmune, metabolic, heart disease, many things. And the question is, what can we do now to fix it? And we think that a big part of the answer is food. And so several years ago when Erica and I came here and started our research group, we actually studied mice as a way of studying the gut microbiome. And the idea was to translate information to humans. So you study mice and then you eventually run a trial in humans. And we started to realize that there just wasn't enough time for this. We wanted to start doing experiments in humans. In studying diet, you actually don't have to go through safety testing. You can just start doing experiments in humans. The problem was we as basic scientists are terrified of humans. And so, because humans are terrifying, and And so we started looking around Stanford, like who could we partner with? And we found Christopher Gardner, and of course, one of the most amazing people in the world for doing dietary interventions. Mark Davis has been a critical part of the Center for Human Microbiome Studies that we've launched basically to develop humans as an experimental model for studying the gut microbiome. And so now we're switching things up where we study humans first and then we reverse translate into mice and try to figure out the mechanism of what we know to be true in humans. And Sean Spencer will talk more about some of this work in just a little bit here. And then what we learn in mice will inform the next trial that we do. And I'll hand it over to Erica now.
Okay, so I'm going to talk a little bit about one of the studies that we did with Christopher Gardner's group, the FIFO study, which stands for Fermented Fiber Food Study. In this study, we took healthy adult individuals, and for the fermented food arm part of it, we had participants eat as much fermented food as they possibly could. They didn't change any other aspects of their diet, just increase the amount of fermented food they were eating. And we do a bunch of microbiome profiling, figuring out what microbes are there and what they're doing. And we also do immune profiling as well to see whether this dietary intervention influences the participants' immune system at all. And we had wonderful people in our lab working on this that I have at the bottom of the slide. And then people in Christopher's group were instrumental in getting the participants to eat the diet as part of the study. So for fermented foods, the instructions were eat as much fermented food as possible, and it could be whatever fermented food they wanted. So a bunch of people stuck to dairy ferments. There were people eating vegetable ferments, kombucha, miso. It was whatever they wanted to do. And participants really did different things. So as a basic scientist, it's a little bit scary to do a study like this because, you know, it's not like everyone drank kombucha. It was like everyone had the fermented food they wanted, and we weren't sure how this was going to work out. But as you'll see, it was actually very informative. So participants increased the amount of fermented food they ate during the study, and that's kudos to Christopher's group to get people to start at, that we're basically starting with 1 serving at most of fermented foods per day, and then during the middle of the study, we're eating upwards of about 6 servings of fermented food per day. When we looked at the diversity of these participants' microbiome, which is just a measure of how many different species they had within their gut, we found that consuming fermented foods was associated with increasing the diversity within their microbiomes. And the reason why this is so interesting is that increased microbiome diversity is generally associated with beneficial markers of health. And we know that in industrialized populations that consume a lot of antibiotics and a lot of processed foods, that in general we see low microbiome diversity. So here was an example where consuming fermented foods increased microbiome diversity of these participants. What about their immune status? When we looked at markers of inflammation, we found many markers of inflammation actually decreased over the course of the study, an indication that these participants were becoming less inflammatory over the course of the study. And this, we think, is important potentially in decreasing the possibility that participants would have a propensity for inflammatory diseases, which are very common in the West, things like metabolic syndrome, obesity, and other autoimmune and inflammatory diseases. We have since completed many studies with Christopher Gardner's group looking at a variety of dietary interventions and asking the question, how do these dietary interventions influence the microbiome, and are these changes in microbiome associated with health outcomes? For example, things like immune status of the host. So we've done low carb, low fat, the fiber fermented I talked about, probiotic, prebiotic studies. We're in the middle of a fiber fermented study in a pregnant cohort to see how this sort of intervention influences the microbiome during pregnancy and also the microbiome that's transmitted to the infant. And then we have other studies on the horizon that we're interested in. I wanted to talk just a teeny bit about a swap meat study that we did in which we had participants eating either real meat or a plant-based meat. And this was a study where participants either ate regular meat for 8 weeks and then switched to plant-based meat, or did the reverse, plant-based meat first and then regular meat. And one of the things that we measured was the amount of TMAO in the serum of these participants. And TMAO is a molecule that is produced by the microbiome, and why we're interested in it is its high levels of TMAO are associated with negative cardiovascular events. So you want TMAO levels to be low. And if you focus on group 2, on the very right, on the left part of that graph, you see when participants are eating meat, red meat, the levels of TMAO in their serum go up during the course of the study. However, when they switch to the plant-based meat, we saw levels of TMAO decreasing, an indication that the plant-based meat doesn't allow the microbiome to produce TMAO. Interestingly, the group that did it the reverse order, when they had the plant-based meat first, didn't make high levels of TMAO like we saw in the other group. However, when they switched to the regular meat, they also seemed to be protected in TMAO levels. So an indication that consuming plant-based meat could also potentially protect in situations where TMAO levels would be higher, for example, eating regular meat. Okay, so finally, I just want to acknowledge members of the Sonnenburg group and, of course, the Gardner group that helps us with all these studies. And then I'm going to turn it over to a member of our lab, Sean Spencer, who's going to talk a little bit about his work within our lab.
All right. Great. Thanks for the invitation to speak today. Very excited to tell you all about my work on fermented foods in the Sonnenburg lab. And really what we're trying to do is create a path to optimizing both the flavor of fermented foods and their functional properties in reducing inflammation. So a key question that informs our work is how dietary intake impacts microbe-host dialogue to regulate our health. I started this line of investigation more than 10 years ago during my PhD work, and really trying to uncover this complex interaction between the immune system, the microbiota, and our diet. And I wrote this perspective about 10 years ago, underlying the complexity. And then I was really excited when I joined the Sonnenburg lab, because really we found or proposed a way to address this almost quantum entanglement of these 3 items. As Justin mentioned and Erica highlighted, we start in humans to generate hypotheses and then test them in mice. And my PhD was primarily in mouse mucosal immunology and biology, and I've taken the human data that we've obtained, generate hypotheses that we test in mice, and then, as was alluded to, we're then designing a new clinical trial to test my findings. It was fermented food, the trial that I'm focused on, and really the main reason that motivated this was that the last bullet point, fermented foods are considered to confer health benefits, but are really not well tested, either in clinical trials prior to ours or mechanistic animal models. And I just want to highlight here that with these data of reduced inflammation, we were still left with the question of a mechanistic underpinning of this observation. Really, the key question, is this a microbiota or host-directed effect? It's likely both. And what component of fermented foods were mediating these observed effects? And so throughout my last few years in the lab, I've been addressing a few questions. What is the chemical and microbial composition of fermented foods? How do fermented foods directly impact host physiology? And how do fermented foods directly impact the microbiome? And so to answer the first question, I leveraged a new platform that was developed in the Sonnenburg lab to interrogate the content of fermented foods. And here, I took all of the fermented foods that participants consumed, and we ran them through this novel mass spec pipeline to identify microbial metabolites contained within fermented foods. In an unsupervised clustering here, you can see that largely the fermented foods group by the type of fermented food. You see for vegetable ferments, yogurt, cheese, kombucha groups with juice, and then a vegan yogurt and kefir at the top. And really what I want to highlight about this figure is that these are the molecules that both convey bioactivity but also flavor, and that these don't necessarily have to be mutually exclusive and are likely synergistic in their bioactivity. So we're looking for ways to optimize both flavor content and bioactivity of the fermented foods. And then in thinking about how to start mouse experiments, we wanted to leverage our data. And so as Erica mentioned, we let them consume whichever fermented foods they wanted. And this allowed us to perform a linear regression on the type of fermented foods and the microbiome diversity that we saw. We saw a couple types of fermented foods pop out to appear uniquely potent in their ability to diversify the microbiome. This was yogurt, and it's called gut shot. This is a basically sauerkraut brine, so it's just a fermented vegetable brine drink. And then, so I took these and then subjected them to an additional method of analysis that looked for short-chain fatty acids. And these are molecules that are particularly well-appreciated components that are made by the microbiome to impact host physiology. And what we saw was quite striking in that there's a big difference in these type of molecules between yogurt and this fermented vegetable brine and kombucha. You see kombucha is mostly acetate, which is also known as vinegar, whereas the yogurt and the fermented vegetable brine were mostly lactate. And so we then leveraged this to do further testing. The point I want to make is that these are large amounts, on highly concentrated amounts, and that participants were consuming tens of grams of lactate per day in fermented foods, potentially. And so, fermented foods are complex, and we wanted to break it down. And we thought that this fermented vegetable brine was a really nice way to do it because it doesn't have a lot of macronutrients in it, essentially none. It doesn't have any significant source of prebiotic fiber. It's really just a source of microbes and their metabolites. And this allowed us to test the individual capacity of either the metabolites or metabolites plus live microbes in conveying an ability to control inflammation. And so what I did is then I gave to mice either the whole intact brine, I sterile filtered it to deliver only the metabolites, and then motivated by our data, I gave lactate only to see the effect of just lactate. And so we colonized germ-free mice with human stool to essentially run the clinical trial in mice. And then the cell type that I looked at is called a T regulatory cell, and you don't need to be an immunologist to understand the relevance. It's just a cell that's very well appreciated in controlling inflammation. And if you have more of these, it leads to less inflammation. And if you have less controlled inflammation, so essentially more inflammation. And what we saw was that by delivering this vegetable brine to the small intestine, you could dramatically augment the regulatory T cell compartment in the small intestine, suggesting that immune regulation was really augmented by this administration of brine. And we saw this primarily in the small intestine. We're very excited to test a sterilized version of the fermented vegetable brine to see if the metabolites themselves were able to convey this phenomenon and potentially independent of any live microbes. And what we saw was kind of surprising, and that the sterile fermented vegetable brine itself was able to convey this effect on mice, this increased immune regulation, suggesting that, at least in this setting, that the live microbes within fermented foods are not conveying this phenotype. And even more interesting to us was when we administered lactate itself, it seemed to convey a similar phenomenon in the small intestine, suggesting that not only are metabolites bioactive, but that lactate is a particularly important bioactive metabolite that can account for this observation as well. And then lastly, we wanted to see, can we use sterile fermented foods and their potential ability to regulate inflammation as a therapeutic? So we reached out to some collaborators who were studying peanut allergy at University of Chicago, and we sensitized mice to make them peanut allergic, and then we treated them with either a control or sterile fermented vegetable brine. So this is just the microbial metabolites from fermented foods. And as you can see in the control group in these gray dots, they had anaphylaxis when exposed to peanuts. And mice, when they anaphylax, lose core body temperature. So you see here that they are anaphylaxing and losing core body temperature. However, when administered the sterile fermented vegetable brine, we didn't observe any anaphylaxis at all, which was quite striking. So we're really interested moving forward to identify key bioactive metabolites, and we're really excited to start to think about making a medical grade fermented food that we could deploy in the clinic and how to start thinking about that, and potentially one that would have potent anti-inflammatory capacity as well as potent gustatory capacity for being delicious. So trying to optimize both of those. So in summary, fermented foods influence host microbe ecology. They do so primarily in the small intestine to promote immune regulation. Fermented food microbial metabolites are likely key, and the metabolites within fermented foods are diverse and distinct. So we're very excited to also explore the diversity of fermented foods. And lastly, lactate is a central component of fermented foods that may be uniquely bioactive. So it's important to have metabolite diversity as well. So a diversity of fermented foods is probably an important component here. With that, I just want to say thank you to everyone, everyone in the Sonnenburg lab and the Sonnenbergs for being such great mentors, and most importantly, my family. Thanks, everyone.
Okay, so let me change my gears a little bit here. I'm more on the engineering side. I'm actually talking about today is about the smart toilets that I've been working on. So basically, they're all biologists here, but I'm an engineer. I'm more on the working on the interface, how we're gonna assess stool directly. So let me start with the precision health. That precision health was conceived by my late mentor, Dr. Sanjeev Samgambhir, who was a chair of radiology. He once mentioned that Stanford Hospital should not celebrate when hospital is full, but we should celebrate when hospital is empty. That's what actually he's envisioning in the future. That's like a part of his plan. His plan to make every kind of the diagnosis can be happening at your home setting. So the precision health starts at the conception of. At the time of conception, and we can do all the risk assessment at the stage of life. Then what we can do is we can provide a customized monitoring tool in order to provide some like a healthcare monitoring of an individual. Then we can feed them into the integrated health portal so that we can track down what's going to be happening in the future. So it's a more proactive and personalized healthcare platform than the precision medicine. So I'm introducing the smart toilet. I think that this is the gateway to the precision health because blood is by far the most well characterized the biofluid that you can utilize a lot of information. But if you're healthy, you're worrying about your needles, right? But naturally, smart toilet because number one and number two, urination and defecation will happen no matter what you're superhuman or not. So what we're trying to do is trying to collect the data from this, and that includes urinalysis, stool analysis, and we're trying to integrate this into the ambient technology, which means it can be a part of Internet of Things. And the later part of this project is the biochemical analysis. We want to see what's the biochemical markers inside, such as blood-based diagnostic or circulating. So in vivo, in vitro. But there's a lot of barriers and challenges for now, so I will explain this later. So this is our first prototype that's introduced in 2020 in Nature Biomedical Engineering. We utilize sensors, optical scanner, which means it's a camera. So it's kind of counterintuitive, but what we're trying to do is trying to scan your excretion in real time. That we want to extract information from the urination and defecation. So first thing that we integrate is urinalysis module. Second is a uroflowmeter. That's analyze your urine speeds and the volume and the duration. Third one is a stool analysis. We're using the machine learning and artificial intelligence to tell you about the morphology of stool and all the defecation analysis too. So first one is urinalysis, extremely simple. We use the urine strip. So we have a mechanical arm to deliver it to the urination site, and we extracted it, the information of the colorimetric change. That's very simple. And second one is a uroflowmetry. So what we're doing is we're trying to measure the volume and the speed of urination. That's the uroflowmeter that's installed in the Stanford Urology Clinic, which where I'm working for. But one of the interesting feature of this is this one has a spinning disc. Spinning disc means it's spinning. But if you want to install it to your toilet, that's going to be a disaster, right? Because, you know, yeah, if you have a spinning disc inside of your toilets, that's... So we propose to use a different methodology. So we're using the two-camera system to capture the urination. And by using the artificial intelligence and computer vision, what we can do is we can extract the volume information out of it. So, and if you use a high-speed camera, what happens is the urine stream is not a stream. It's a droplet. It's a series of droplets. So what we do is we characterize a droplet, size of droplet. So we we reconstruct it into the 2.5D structure, so we can measure it, and it has a really good correlation with what we can measure. And the third, so you must be interested in the stool analysis. Stool, of course, is very important, and we doctors, I think Sean knows about this, right? This is the Bristol Stool Foam Scale that tells you about how your stool looks like, right? And we utilize this card, but the thing is, if one of the doctor's problem is that the patient may have a recollection bias. Because let me ask you a question. How's your stool today? Right? Nobody can remember it correctly. So what we actually trying to measure, trying to use is objective manner to analyze a stool morphology. We're using Google Inception V3, which is a machine learning module that's publicly available. And this is a publicly available module that utilizes a flower image, but we replace it to the stool image. And this is how hardware looks like. It's extremely simple. We have one Raspberry Pi computer with a camera, which is downward, not upward. And we have LED strips that's triggered by the sensor, the seating sensor. Whenever you sit, then it actually turns on the lights. So it looks like this. This one is installed in the clock center, was used to installed in the clock center. So this is a real-time analysis of stool, so defecated. So we have AI is actually capturing where the stool is. So we can measure the volume, we can measure the color, we can measure the duration of the defecation. And this is very unique analysis tool right now. And we have 11 participants who generated 55 defecation events on this toilet. So this is the result. As you can see, so we have 55 defecation events with 5 male, 6 female, and our match rate is 85%. And this is the longitudinal monitoring. As you can see, the red participants consistently have some kind of constipated stool, but the participant never realized participant has this constipated stool at all. In that case, we may actually recommend a stool softener or psyllium. Or the other, if you see the cyan color and the blue color, they are coupled. And as you can see, they're sharing their stool morphology almost exactly same. I'm actually wondering, they may actually share the food together, right? It may actually result in this. They actually have some kind of diarrhea over there, so we may actually recommend the anti-diarrhea medication for both of them. So this is industry-grade prototype, which is installed in my house. So, and it's a monitor every excretory pattern from almost 87 days, and I have 800 events, 600 urination, 200 defecation events. So, most importantly, we're working with NASA because NASA's next 10 years plan is to send people to Mars, right? Sending people to Mars usually takes more than a 6 months journey. So which means space crew member will be facing to very extremely hostile working environments, right? It has a microgravity, radiation, everything. So we're proposing space precision health. We want to proactively monitor the spacecraft crew members' health. So one of the way is to utilize a smart toilet with a microbiome, right? So microbiome has a response to the immune system. So if we have those kind of system, we can install that into the spacecraft. Yes, I have one more minute. So this is another interesting factor. And the second way is we can deploy this module to the smart cities. Because as you are aware of the wastewater study, they're looking at the COVID-19 spread, right? So if we have those kind of analytical capability, then we can use our toilet as the diagnosis sentinel. So last but not least, I think one of the challenges is a psychological barrier. Even I mentioned to you, you laugh at it, right? So people may think that this is funny, but it's a very important subject that I'm actually working on. And there's a social taboo too. I submitted a proposal to NSF last year, and I got a really horrible score. Majorly, they're talking about the taboo nature of the human excreta. So I have to address that. And there's an engineering challenge, there's an economic justification. Most importantly, there's an ethical consideration because we're looking at your very private events, right? And how can we share that information with you or with your doctor? What's a proper boundary? So we're working with a bioethics specialist at Stanford, Dr. David Magnus, and Dr. Nicole Martinez. And thank you so much for having me today. So he's my mentor for the last 7 years. He was the original conceiver of this smart toilet almost 20 years ago. Thank you.
All right, well, that's a hard act to follow. But I'll try. My name is Emi Bhatt. I'm a physician scientist here at Stanford, and I am really, really interested in how we can precisely study the gut microbiome to improve outcomes in patients with diseases. So these are my disclosures, none of which are particularly relevant for today's presentation. So I study patients that have hematologic malignancies. By show of hands, if you feel comfortable doing so, how many of you know somebody or have personally had cancer? That's pretty much everybody. How about you know somebody who's had a hematologic malignancy, leukemia or lymphoma? Look around, that's probably about a third of us. How about somebody who's undergone a stem cell transplant or bone marrow transplant? Still a good number. So while you may think that bone marrow transplant or hematopoietic cell transplant is very rare, it's actually surprisingly common. And it's a treatment that we use to cure people who have otherwise incurable hematologic malignancies. One problem that I and others have noted as physicians in this space is that our patients, unfortunately, have a very, very high risk of dying after transplantation. Indeed, about 20 to 30% of individuals who undergo a stem cell transplantation will die in the first year after transplant. And these are actually some of the real statistics for the Center for International Blood and Marrow Transplant. What you can see is that in the first 100 days after transplant, about 3/4 of the people who die don't die of a relapse of their cancer. Rather, they die of something else. And even if we look at people who make it out to day 100, about half of them who pass away of something other than their cancer. Almost every one of these non-relapse related mortality events has been, by previous research, been linked to the gut microbiome. So my big question is, how might we be able to use our understanding of the microbiome and modulate the microbiome, be it by food or by medications or other interventions, to decrease the number of people who die of non-relapse related mortality? So to do this, my lab has become really, really interested in measuring what matters in the microbiome. You heard from Justin and Erica and Sean and Sungmin about different ways that we can understand what's going on in our gut, be it directly visualizing stool samples or characterizing the impact of things like fermented foods on who's there in the microbiome. By doing these types of analyses, what we've been able to demonstrate in our lab is over there, just a mile away in Stanford Hospital, about 40% of the patients who experience bloodstream infections who are undergoing transplantation, that bacteria came from their gut. It didn't come from somewhere else in the hospital. It didn't come from a nurse or a doctor. It didn't come from exposure to a dirty sink. It actually came from their own gut. And the vast majority of those individuals, they walked into the hospital with that pathogen. Which has led us to ask the question, well, if we are all walking around with our greatest enemies just sitting there in our guts, how can we prevent them from getting into our bloodstream? Might we be able to crowd them out? To address this, we and others have been experimenting both with fibers as well as specific antibiotics and other types of interventions to try and modulate the gut microbiome to improve outcomes. Another area of interest for us is trying to understand this very interesting observation, which is that a subset of people who get immunotherapy for the treatment of cancer will get completely cured. This is actually one of the biggest advances that has happened in cancer medicine in the last 10 years. It took people who otherwise were going to die of their cancer and it turned them into people who had been cured. Now, the problem is it only works for some people and it doesn't work for others, and we have a really hard time of predicting who's going to benefit and who won't. Furthermore, we'd love to be able to take somebody who is going to be a non-responder and turn them into a responder. So to date, lots of researchers over the course of the last 5 years have demonstrated that the composition of the gut microbiome, so in this cartoon, whether you have more of the yellow bacteria compared to the red and blue, may be associated with whether you respond or not. The problem is every single study that has reported on this reports a different bacteria being important, which is quite confusing. So what we've asked is maybe it's not a specific bacteria that determines whether or not you respond to immunotherapy, but rather how many bacteria you have in your gut. And so this has been an area of great interest for us in the lab as we try to measure the absolute concentration of bacteria in your gut, with the idea that we might be able to modulate that by things like eating foods that will increase the absolute abundance of bacteria there. Not only are we interested in bacteria, but we're also interested in phages. Everybody knows about viruses, right? Viruses infect us. A virus just like shut down the world for a few years. And it turns out that there are also viruses that infect bacteria. Those viruses are called phages. And it turns out that they are extremely numerous. There are probably as many phages in your body as there are bacteria, which means that each of you is carrying around 1 trillion of them. Yet, until recently, we actually had very little visibility into who these phages were and what they do. And so we have been developing tools to be able to measure and study these phages more comprehensively, so we can understand how these phages, which really are controlling bacteria, impact our health. So you already saw this beautiful image, but you saw it flipped around 180 degrees of the cross-section of a colon. We in the field have been interested in asking who's there in the microbiome, both bacteria and phages. We're interested in what they make, not only small molecules like lactate, which Sean brought up, but also small proteins. We're interested in how these proteins and small molecules actually traverse that green layer, which is the mucus between the bacterial component and the human cell component of the gut, and communicate to us. So we also are very interested in deciphering this microbial communication. How do these molecules and proteins that microbes make enable them to speak within species, between species, so to each other, and even directly to us, between domains of life? If we can understand this type of communication, then we can hopefully intervene and change that conversation. So with that, I'll close by saying we know that there are all kinds of important things that are happening in our gut, ranging from barrier function to immune modulation, infection and nutrient extraction. We know how to measure who's there, and more we're able to figure out what these microbes can make. Our goal over the next several years is to figure out how these two interact and to use this information to design interventions that range from food to medications to try and improve host health. If we can precisely measure microbes, maybe we can enable precision medicine. So with that, I want to thank my totally outstanding lab, our great collaborators, and we're open for questions.
I got one mic here. Oh, you have two up there. Jennifer's getting one there. How many of you are ready to sign up for the poop study right now? Hands up. Look, yes. Okay. We should be recruiting right now. All right. And I think I had a question back here. Who had a hand up?
I would like to thank you for all you do and for all your research. I would like to ask the effect of antibiotics to microbiome, because, yeah, just if somebody can answer.
Okay, so thank you for the question. It was about, what do antibiotics do to the microbiome? Fundamentally, antibiotics are really, really blunt tools. They kill not only the pathogens that they are intended to kill, but they usually have a lot of off-target effects as well. And so, from a physician perspective, it's really important to keep in mind that if people need antibiotics to treat an infection, they certainly should take them. But what we find is that there's a huge amount of antibiotic overuse, and antibiotics can have a lot of unintended consequences on the gut microbiome. One of the things that they can do is they can actually kill a lot of the healthy commensals in our gut microbiome, thereby making space for organisms that can be pathogenic and eventually do things like cross into the bloodstream and cause infections. There are times in which giving antibiotics can be strategic for preventing infections. But I think everything that we've learned to date suggests that the antibiotics that we use, and even medications that are not antibiotics, such as chemotherapies and other drugs, can have unintended consequences on the gut microbiome.
Hi, thank you. That was great. My question is for the Sonnenburg lab. I'm just curious if you've looked into like a minimum effective dose, appreciating that fermented foods aren't necessarily everywhere, and what your thoughts are on that.
Yeah, so in our study, we wanted them to eat as much as possible, so they ended up eating about 6 servings a day, which, for the average American that eats 1 or less servings a day sounds pretty overwhelming. But actually, the serving size is not as big as you would think. So like a 6-ounce glass of kombucha is considered a serving. So most bottles of kombucha are at least 2 servings or maybe more. So, if you have some yogurt for breakfast, a bottle of kombucha, you add a little bit of sauerkraut with your salad, maybe you have a glass of kefir at dinner, you can get to 6 servings like pretty easily. However, in the second part of the study, after they were eating 6 servings a day, we provided this choice period where we told participants, just, do whatever you want. You can go back to your old diet, you can continue eating 6 servings, just whatever you're comfortable with. And what ended up happening was most participants landed around 2 to 3 servings of fermented food per day. And during that 4-week choice period where they were only eating 2 or 3 servings, we still saw increasing microbiome diversity happening at that time, which is an indication that 6 servings maybe isn't required, that maybe just 2 or 3 servings per day might be enough. But that's why we're interested in doing additional studies, and then the mouse model work will help us with that dosing effect. But it does seem like even just maybe 2 or 3 servings a day could be beneficial.
So talking more about antibiotics, I'm curious, I've been wondering in terms of animals that have been raised with antibiotics, has that been seen to have an effect in the microbiome?
Okay, so I'll start. Long story short, there's a lot of suspicion that actually the antibiotic exposure both in livestock as well as the runoff from livestock that often makes it into our agricultural supply actually is making it into our diets, and therefore giving us exposure to kind of sub-therapeutic doses of antibiotics, which may actually be to blame for the very dramatic rise in antimicrobial resistance that we're seeing in pathogens throughout the world. There's also the suspicion that this might be having small but meaningful effects in changing the gut microbiome, especially in individuals who are exposed to that food source. So it's an area where there's a lot of speculation. It's also a lot of lobbying. So it's an incredibly complicated space and an important question.
But I would say that for the studies that I've seen, it doesn't appear that antibiotics that are used to treat livestock make it into the meat in sufficient levels to impact our own microbiome. It doesn't mean that it's a good practice, but that's the data I've seen.
Yes, I believe Steve Jobs said that the Apple Watch was going to be the most personal piece of technology that a human would wear. And I believe I heard on NPR this discussion about the smart toilet, and you had a camera that did point up. Could you just quickly speak to precision medicine in identifying each member of the household with the camera that pointed up?
Thank you so much for a very important question. So, because your toilet will be shared by your roommates, housemate, family members, right? So for precision health, I think it has to be very personalized. Otherwise, there's going to be a mixed up with information, right? You don't want to have. So our eventual goal is to integrate those information with EMR or EHR system so that the physician can take a look at what the stool pattern looks like. So first, we envisioned that I think the very straightforward way is to integrate the fingerprint scanner onto your flush toilet. I mean the flush. So whenever you flush it, then your all generated data will be annotated with your personal identity. But there's some glitch because some people may not actually flush at the right time, right? So I have some example because I have a 1-year-old boy, so I don't want to wake him up during the night. Then sometimes I skip the flush, and the next day my wife can flush instead of me, right? In that sense, there's going to be some kind of mix-up possibility. So, So there's a second way to analyze people's identity that's known as anal prints. So Dr. Sanjeev Sangambir was conceiving this idea that human anus is very unique to a person, and it has one orifice with 35 to 37 creases around it. But it's very unique as your fingerprint did. So we utilize that as a biometric identifier. So we use some patient data to, I think it has more than a 95% accuracy that we can trace back. And interestingly, we can actually evolve those systems to analyze any kind of anus-related disease too. Because anything that's related to your anus is assessed by visual assessment. Basically, doctor has to see it, and that actually gives give you the final diagnosis. So what we can do is we can train the machine to analyze like anal fissures, anal cancers, hemorrhoids such as. And interestingly, we actually found some profound application toward to the IBD patient who usually have a rectum symptom a lot. So we're developing it, but as I mentioned, this is very related to the privacy and ethical consideration. Is it right to picture someone's anus? So we're working on it. Thank you so much.
Okay, do you have one more question here or two? Okay, go ahead. You've had it.
I was wondering if you could speak to the efficacy of fermented foods versus probiotic pills and what you would tell someone who wants to just pop a probiotic versus actually incorporate fermented foods into their diet.
Yeah, that's a great question, and we actually did a probiotic study. It was a different cohort. It wasn't exactly the same as the fermented food study, but we were thinking, when we saw this great signal with the fermented food, increasing microbiome diversity, decreasing markers of inflammation, that one of the obvious things to try was a probiotic supplement to see if we would see something similar. And surprisingly, we didn't. There was people taking the probiotic supplement. It was a placebo-controlled trial, so it was really done well. people taking the probiotic, no change in microbiome diversity over the course of the study, no difference in immune status markers over the course of the study. And initially we were really surprised, and then Sean started doing his experiments with the sterile fermented foods, and it does appear that the microbial compounds within the fermented food, things like lactate and these other metabolites, might be a really important part of the fermented food outcome that we saw. And so by taking the probiotic, you're only taking the bacteria. You're missing out on all the compounds that those microbes are creating within the food. So it doesn't mean that there may not be indications or specific situations where a probiotic supplement would be beneficial, but at least in this the studies that we did, the main signal was with the fermented food and not the probiotic supplement.
Thank you. My question, first of all, thank you, great talks, very interesting field. My question is, does the gut microbiome have any role to play in brain development and in general in mental health? And are you also focusing on that? Thanks.
Very good question. There is a lot of compelling mouse model data to suggest this is the fact, that the microbiota influences development in all parts of the body. There was a compelling human clinical trial recently actually centered around anxiety and fermented foods and fiber. So there was a group in Ireland that did a very similar intervention to the fiber and fermented food. They actually combined fiber and fermented food in individuals that self-reported anxiety. And they tracked their anxiety over the course of this interventional trial and actually showed in this randomized trial that those randomized to the fiber and fermented food had reduced levels of anxiety. Again, the mechanistic connection between the two is not yet established, but this is compelling human data to suggest that there may be a connection, and our job in the field is to try and figure this out in the future.
Okay, there's no huge line here, so if anybody had we started a line? Yeah? Okay, the line has started. Round of applause. You guys, can you imagine